请来刘鼎给我们做特别主持人，然后下面就由他来介绍一下我们的 speaker。好
，OK， 因为 speaker 要用英文讲，我又用英文来介绍了。Okay, let's get started. Welcome everyone, and please allow me to introduce today's speaker, Dr. Zhang Ying. We call her Amanda. Ying got her bachelor's degree at Nanjing University, and then she went to Zhejiang University for her PhD training at Li Xiaoming Lab on the research of cholinergic systems in Rett syndrome. Today, she will introduce her postdoc work from Guo Pingfeng's lab at MIT on the functions of thalamus nucleus and their contribution to autism and schizophrenia. Ying is a best friend of me at Boston. We know each other for many years. She's a highly talented and hardworking neurophysiologist. Let's welcome Ying. All yours.
Okay, thank you for the introduction, and thank you everyone for coming. It is my great honor to share our recent paper here. This project is about the convergent function of anterior thalamic nuclei in neuropsychiatric disease models, and it is co-led by Dhiraj Roy and me. Dhiraj is graduate from MIT and studied memory engrams in Tonigawa Susumu lab. He enjoyed a festival of CNS papers, and now he's also here. He will join us in the discussion part of today. Thank you, Dhiraj. I'll buy you coffee next week. Okay, why study anterior thalamic nuclei? Research so far has been extremely important in understanding memory formation, but input circuit regulating the hippocampus and renal network has received much less attention. ATN has been reported to have reciprocal connectivity with the hippocampal cortical network. Secondly, degeneration of ATN is found in many disorders, suggesting a potential role of ATN in Alzheimer's disease and schizophrenia. Third, ATN is activated in memory task, and lesion studies in Rett has found that ATN is crucial for spatial navigation and cognition. ATN is a less studied brain region which contains 3 subdivisions, AD, AV, and AM, with their individual role in normal and disease state has never been investigated. So why us? Prior studies from Dhiraj used the brain-wide engram mapping directed him to AD thalamus, which is among the top of the list. With this in mind, he joined Guoping Lab and invited me in this exciting journey. We started by determining whether specific molecular marker could be identified within ATN. Taking advantage of the RNA-seq data from Steve McCarr, we focused on 11 excitatory clusters in mouse thalamus and noticed that one cluster has the highest C1QR2 gene expression. Our staining experiment showed that C1QR2 is specifically expressed in AD within ATN, and this expression pattern is also conserved in marmoset. Since we found the molecular marker, one direction is to generate Cray-Mouse line, which would take around 1 year. Both Dhiraj and I would consider that as a great idea if we have no other choice. It is knowing that ATM mailing project to PRESAP and RSC area. Through CTB retrograde tracing, we found that RSC received projections from both AD and AV, while injection of CTB in PRESAP only label AD neurons, but not AV neuron. This experiment not only revealed the differential projection pattern of AD and AV, it also gave us a hint that through manipulation of this circuit, we may be able to target AD and AV areas specifically. By injecting a retrograde Cre virus in pre-sub and Cre-dependent rabies in AD and AV, we revealed differential input to AD and AV area. Given that AD and AV have distinct molecular markers and connectivity patterns, we then compared their electrophysiology properties. We injected rabies expressing GFP in pre-sub, which could give us a clean boundary between AD and AV. Within AD, GFP positive and GFP negative neurons had similar properties. However, we observed a striking difference between AD and AV. These experiments revealed distinct properties between AD and AV thalamus. So our next question is, what is the role of these ATN subdivisions in memory process? We injected DGL Cre virus in pre-sub combined with Cre-dependent HM40I virus in AD and found that inhibition of AD during training impaired the long-term memory recall. However, their freezing behavior during training and in neutral context was unchanged. In search of the cellular correlates of memory encoding in AD, we found that the frequency of mini-EPSC was increased in AD after training, which is correlated with the increased number of CFOS positive neurons in AD after training. AD could project both to pre-sub and RSC. Through a series of experiments, we found that the projection from AD to RSC, but not AD to pre-sub, is involved in memory encoding. Optogenetic terminal manipulation of AD-RSC circuit suggested that this circuit is not only necessary, but also sufficient to enhance long-term memory recall. Hippocampus has been recognized as the most famous brain region for contextual memory. We found that AD inhibition blocked the training-induced enhancement of CFOS ensembles in CA1. This suggested that AD could contribute to hippocampal activity. So our next question is, how? What is the circuit basis of this observation? Entorhinal cortex serve as a mirror input to hippocampus. So does RSC neurons that receive projection from AD could project to entorhinal cortex? We injected a retrograde Cre in EC combined with Cre-dependent rabies in RSC and found that AD neurons, but not AD neurons compared to AV neurons, project to EC projecting RSC neurons. We then injected anterograde Cre virus in ATN, Cre-dependent arch in RSC, and implanted fibers in EC. Direct inhibition of ATN RSC EC circuits during encoding impaired long-term memory recall. So, using a circuit-related manipulation, we found that AD is crucial for contextual encoding through projecting to RSC. This observation was further supported by optogenetic terminal inhibition of C1QR2 Cre mice. Since AV also project to RSC, we wanted to know whether they play any role in CFC memory. We injected DGL Cre in pre-sub and Cre of NPHR in ATN area, which give us a specific label in AV. Different from AD, inhibition of AV cell body or AV-RSC circuit during training had no effect on long-term memory recall. However, these mice displayed a robust generalization in the neutral context test. We next quantified the activated examples in RSC while inhibit AV-RSC circuit. Strikingly, AV-RSC inhibited mice showed increased C-force examples in RSC, which suggested the possible involvement of inhibitory neurons in RSC. To pursue this possibility, we injected Cre-dependent rabies in RSC of PV-Cre, SST-Cre, and VIP-Cre mice, and found that AV mainly project to PV and VIP inhibitory neurons in RSC. So, do these cell types play a role in AV-RSC circuit in terms of memory specificity? We inhibited AV-RSC circuit while simultaneously activate PV or VIP neurons in RSC. We found that activation of VIP but not PV prevented the generalization phenotype with AV-RSC inhibition. This experiment suggested that AV's role in specificity requires RSC VIP neuron. So, in the basic science part, to summarize, we uncovered the role of convergent ATN circuits in memory. AD contributes to contextual encoding through AD, RSC, EC circuits, and AV is crucial for memory specificity through RSC VIP inhibitory neurons. So, does ATN play any role in neuropsychiatric disorders? Guopingfeng Lab has a long history of autism study. PACHD1 is a risk gene for autism, and our lab's previous work with PACHD1 knockout mice showed that TRN is responsible for all other behavior deficits except cognition deficit. Our in situ data revealed the selective expression of PACHD1 in AD. So, is it possible that AD is responsible for cognition deficits in PACHD1 knockout mice? We extended to more autism risk genes from Safari database and noticed more genes had clear AD expression. Cognition deficit is also a core phenotype with schizophrenia. We went through the top 10 risk genes for schizophrenia and noticed that 3 genes exhibited high expression in AD. To test whether AD dysfunction plays a role in memory deficit in PACHD1 model, using a CRISPR-Cas9 approach, we selectively knocked down PACHD1 from AD and found a long-term memory recall deficit in field conditioning test, together with a working memory deficit in T-maze test. We then performed knockdown of all other seven ASD and schizophrenia risk genes listed in my previous slides, and found 4 out of 7 exhibited the same behavior deficit as PACHD1. This is an example for ASD risk gene, YW-HEC and HEC1. We then wanted to examine how PACHD1 alters AD neuronal properties. Using slice recordings, we found a decrease in action potential half-wings, which correlated with an increase in the excitability of AD neurons. This neuronal excitability increase in AD is also observed in YW-HEC and HEC1 knockdown in AD, revealing a convergent cellular phenotypes across disease models. We next wanted to identify the molecular mechanisms underlying hyperexcitability in AD neurons across different disease models. We focused on ion channels that are necessary for maintaining action potential thresholds and also half-winds. And the second criteria is these channels should be expressed in thalamus area. Through direct recording of different ion channels, we found that Cur 2.1 current is decreased in YW-HEC and HEC1 knockdown mice, while two other different cathode currents, CAV 2.1 and 2.2, are decreased in PACH-D1 and HEC1 knockdown mice. Even though through different ion channels, selective knockdown of different disease risk genes from AD all led to neuronal hyperexcitability and cognition deficit. We then would like to test, would it be sufficient to rescue memory deficit if we treat the neuronal hyperexcitability? We next developed a dose-dependent chemogenetic approach to normalize the excitability of AD neurons and found that if we bring the excitability back to physiological level, the memory deficit could be rescued. We further demonstrated that this strategy could also apply to YW-HEC and HEC1 knockdown mice. So to summarize, we find a molecular marker for AD thalamus, and we generated C1Q1 mice and also developed a circuit-based manipulation method for the manipulation of AD-AV, and found that AD is crucial for memory encoding, while AV regulates memory specificity. We then find out that knockdown of several ASD and schizophrenia risk genes in AD led to cognitive deficits. 3 different knockdown models exhibited AD neuronal hyperexcitability. Then we showed that rescue of hyperexcitability is sufficient to rescue cognitive behaviors across disease models. So we find cellular, circuit, and behavior convergence underlying cognitive deficits in a subset of neuropsychiatric disease models. So I'd like to thank my collaborator, Dheeraj Roy, and he initiated the project, and we really enjoyed the journey. We hope we could be the top 1 team in the world. We did everything, we did every experiment together with Dheeraj Roy. And Tomomi is a research scientist in Gopin Lab, and he is the expert in molecular experiment. He created the C1Q1 mice for us and also developed different CRISPR-Cas9 virus for us. And Sunuk is also a research scientist here. He is an expert in vivo electrophysiology. I didn't show his data here because I'm afraid that we will not be able to go to bed early tonight. And Yuan and Nick performed a lot of surgery and also staining for us. Wing, King, Ming, Hannah, and Juliana are the undergrads in our lab. They have done a lot of analysis and behavior experiments for us. Chen developed the virus for us. And Kater also did a lot of surgery for us. Jay Dong from Tufts provided the Cas9 plasmid for us. Heather Yan from MIT provided all the rabies virus. RP and Steve gave us the earlier access of the RNA-seq data, which made the C1Q1 mice possible. And special thanks to Guoping. Yeah, that's all. Any questions?
Okay, I think you're just ran a bit more faster. That you should elaborate more details, but it's okay. We can start the Q&A part. We can explain more details in the question part.
Yeah. Whoa, so quick.
Yeah, maybe you're a bit nervous. Let me get started. So the first question is, can you explain more the details about the behavior paradigm you use to measure the memory? I think there are several, right? There is a long-term memory assay and a working memory assay, and what are they?
Yes, you mean these slides?
Yeah, you mentioned multiple times the long-term LTM test and the other test. Can you explain more? How do you do the experiment? And what's the phenotype of the animal? Yeah.
Yes. In this experiment, we inhibit the AD neurons during the training time. And then 1 day later, we did a long-term memory test and found the behavior deficit. And in addition to that, we also inhibited during the, right after training, and also during the long-term memory test. There's no behavior deficit if we do other manipulations.
Okay, so this is a fear conditioning, right?
Yes.
So what is the result of the test? Oh, it's the freezing, I see.
Yeah, freezing.
It's like you only play the tone and to see the freezing. It's a
we didn't play the tone. There's no behavior deficit in the tone field memory. We did the contextual based field memory.
I see. And what about the teammates working memory task?
Yeah, teammates working memory test, we did 2 versions. One, the delay time is, one it is 10-second delay, and another is 16-second delay, which is more demanding. So we find behavior deficit with the more demanding teammates test.
I see. It's an alternative teammates, right? This time left and that time right. So there is a delay in between. Okay, yeah. Okay, other questions? There is one in the chat.
The chat?
I read the paper and wonder why regular dose of C21 doesn't work.
Regular dose. I see. It's because here. Let's go through there. So this one is the regular C21 dose, and it inhibited the neurons, AD neurons, too much. That our hypothesis is, if the effect of CT1 is too strong, that the neurons could not have like our hypothesis is that AD neurons could get more excited during the training, which generated the how to say it? Draj, could you help me?
Hi, can you hear me?
Yes.
Yeah.
Okay. We don't know who that is, but Nova 7. Good question. Let's quickly fix this. So the standard dose of C21, okay, whatever that may be, 2 mg per kg or 5 mg per kg. The purpose when you use HM4DI is to inhibit neurons. So you inhibit these neurons way past the physiological range. So when we started these experiments, we noticed that just using standard dose, there's no rescue. And then when you go and patch, you go and see that the excitability is actually not anywhere close to what a normal AD neuron should be. Only at that point, we started to go and titrate the dose of C21 to find one where A, can you get the excitability closer to what a normal AD neuron should look like in these knockdown animals? And only then, B, do behavior and check, can you rescue it? So I think your question, if I'm understanding it correctly, the C21 works completely fine. It's working so well that it inhibits it exactly how it should. The purpose in this case is you don't want to inhibit it that much. You want to bring it back to baseline, right? Or at least the normal range. So I hope that answers your question.
Okay, feel free to ask questions either in English or Chinese.
Hi, very interesting talk. I missed the introduction part. So you mentioned a lot like AD, AV, RC, EC. So I'm just a little bit confused about this. What's the connections? What's kind of anatomic connections between the different cortical regions and how they contribute to the cognitive performance?
Raj, could you answer this question?
Sure. How about I take the English ones? Okay. So let's quickly just highlight. So in general, in the ATN, right, whether it's the dorsal part, the ventral part, or the medial part, all three of them are well known to interact with spatial learning and memory structures. Now, what does that include? For example, retrosplenial cortex, very well known having reciprocal connections with, for example, hippocampus, parietal cortex, cingulate cortex. In some cases, the output of the hippocampus, like presubiculum, parasubiculum, postsubiculum, okay? So this is already well known. In our case, particularly, if you just focus on AD, the two strongest outputs, not only from us, from all literature, so you don't need to get any special credit here, is one to presubiculum, sitting right after subiculum in the primary hippocampal formation, and then the other is to RSC, right? And in the case of AV, the strongest output is directly to RSC. So for starters, what we basically show is that both AD and AV going to RSC seem to contribute to at least this contextual learning and memory paradigm that we used. Very simple, field conditioning, not very hard. AD to RSC, we think is involved in the actual encoding window. And what encoding? It has to be context encoding, because this is well known to be a head direction brain region. So this brain region fires every time you look at something contextual. Even if it's in the dark, if there's low light, AD neuron will fire, right? On the other hand, AV we know a little bit less about, but our findings suggest that AV regulates the inputs from AD to RSC. So the way you think about an inhibitory neuron in any cortical circuit, we think about AV inputs. So it's kind of cool. You have two neighboring subdivisions of ATN. One seems to be the major driver, encoding, firing when this animal is learning context equals scary. The other one, its neighbor, is 100% excitatory and there is no intercommunication, projects to the exact same cortical region, RSC, only this time controls how active the RSC neurons are that receive AD input, okay? That's how we of course, I'm summarizing a number of experiments here, but that's why we ended with AD to RSC for encoding, for contextual encoding, AV to RSC for memory specificity, because the animals generalize if you take away AV input. Now about the AD RSC, I'm not done. Give me one more minute and I think it'll help. Now you asked about AD, RSC and EC. Very good question. When you think about the brain, right? We're focused on the AD and AV, and then AD projects to RSC. I think it's not fair for readers like all of us, for example, to assume how does RSC communicate this information to, let's say, hippocampal nuclei, right? So we don't want to leave you hanging almost, unknown, a question mark. So when you think about RSC, there are many routes from RSC to hippocampus. RSC can go directly to CA1, to subiculum, presubiculum, or to entorhinal cortex. So Ying didn't show you each of the negative experiments here, but long story short, we linked this memory encoding function for AD neurons that project to RSC to this EC, one intermediate nucleus. So that's the connection. This memory encoding function of AD to RSC, when you wonder, how does this RSC information reach or alter hippocampal activity? It's not directly. It's through entorhinal cortex, and entorhinal cortex, as most of you or some of you may know, projects to either dentate gyrus or CA3 or CA1, right? It's the major input to hippocampus. So I don't know if that helps a little bit, but that's the connection of why the two circuits, because we think that's our encoding circuit, at least from ATN, for a contextual memory experience.
Yeah, that's good explanation, but I'm not to work with thalamus, so I'm not really familiar with the anatomic structure of thalamus. You mentioned about the AV and AD. Is that two nucleus in the thalamus?
Oh yes, sorry. I apologize. So yeah, so the ATN is in the anterior part of the thalamus. So where are we talking? If you think about cingulate cortex, or maybe there are more cortical people here, or retrosplenial cortex, right below it, before the hippocampus shows up in a coronal section of a mouse brain. I hope I'm talking to mostly mouse researchers. It's right below the corpus callosum, basically. Okay. Corpus callosum, ventricles, right below. A cute little 3 structures in one big unit, and they all are excited to the neurons 100%. And the cool part is they all only project to the, outside of the thalamus. So there's no intercommunication. So and I think a lot of people didn't have a way to manipulate it. So the take-home message is not that ATN in general is important for learning and memory. If anyone told you that, it's not surprising. This paper is not the first. But if people told you, what the hell are each of these subdivisions doing? Can we start giving some more information on which circuits do what? This paper may be the first.
I just curious about it because I didn't know, I don't have any background for what kind of cortical circuit contribute to cognitive performance. I just want maybe take this advantage to understand a little bit. So you mentioned about the cingulate cortex. So is there any direct projection from the cingulate cortex to the thalamus, and how this connection then maybe to the entorhinal cortex and maybe to the RSC or some hippocampus? So do you have a simple diagram to show that how this different cortical regions, they are integrated in the cognitive performance? Do you have a more simple cartoons to show background introduction?
Yeah, I see. To be honest, the simplest cartoon is right now on the slide.
Okay.
If you just think about, if you want any simpler, we would have to hide almost half the brain, because there are way too many connections, and I understand, right? There are almost 100 arrows. If you think about retrosplenial cortex, entorhinal cortex, cingulate, these structures, not only for thalamus but for most of whatever brain regions you guys study, it talks to them, either unidirectional or bidirectional. So yeah, I apologize. If we actually made a cartoon, it would take us 2 hours to just go through the cartoon. But this is a very simple idea that think of all these cortical structures, RSC, cingulate, some parts of posterior PFC, mainly talking to ATN in the thalamus, hippocampus, and then parahippocampal structures right outside. And if people have to draw a little picture, this is basically it right here. ATN, RSC, hippocampus, cortical sites, parietal, occipital, cingulate.
So the lights up regions in the diagram is all the regions related to the cognitive performance. Is that right?
At least these are, the very famous, among the very famous ones, correct.
Okay, so you think the blue one, the light blue, that's which cortical region? That's a cingulate cortex? I think it's not cingulate cortex. That's adjacent to cingulate cortex. So which one is the first?
RSC.
That's RSC. Okay.
RSC. Yes, in blue. Yeah, retrosplenial cortex, right? And people think of it as it allows you to make a map of a room, right? or shopping mall, or any new environment you go to. Yes, that's what retrosplenial is supposed to do. When you enter a seminar room or a lunch room, you use cues in the environment and you make a map, right, where you're sitting in 3D compared. That's why I think people like to draw this as a central hub. It uses information from thalamic nuclei, from cortical sites, from hippocampus to make this world map, contextually at least.
So also the striatum also involved in this cognitive performance?
To be honest, in general, people think it has a minor role, at least in the traditional, context is fearful way, a very small role, if any. I'm being very kind. Hopefully there are no striatum people here, but yeah.
So which one you think is the most relevant to the most key regulator for the cognitive performance? Is the thalamus you are studying, or other cortical region you think is more important?
I mean, to be honest, I would love to say that ATN is the most important, but that would be extremely early. I think most people will say the famous word hippocampus. That's why when you Google hippocampus, it does everything. Basically everything you can think of in memory. Even things that we don't know it can do, right? So I think people agree from a lot of literature, right, that hippocampus does something about spatial, contextual memory encoding, and that's the type of memory we're studying. So I have to be careful. Not every memory, but at least spatial, contextual memory. So for these types of memories, the most important, hippocampus by far.
Okay.
That's why if you look in this study, we try our best to link how does ATN regulate either RST directly or hippocampal structures.
Okay, can I ask you later, because maybe I'll take too much time, so I just leave the question to other people. Maybe we can talk later. Yeah, I do have a lot of questions for you.
Great. I mean, since I'm speaking, there's one question in the chat. Might as well take it. It's the same, Nova 7. What is the function of AD pre-sub? Very good question. We have no idea. If you do, please share. But what I can tell you is the following. Presubiculum itself, remember I told you that anterodorsal thalamus actually became popular in the literature for head direction. So every time your head turns in three-dimensional space, these neurons code for the direction your head is relative to your body. Cool. Presubiculum is the second highest brain region in terms of percent of neurons that have this head direction firing. So we have not proved this because we didn't have a beautiful head direction task, but a very good guess is that AD to pre-sub is involved in head direction. And why can I guess this? If you look at the strength of this circuit, it's almost 30 times stronger than AD to RST, right? And what does it tell you? Very few circuits are at this, let's say hundreds of picoamp range in a slice. So most likely these are doing something to do with sensory-driven processing. I don't have, convincing evidence yet, but as a very good guess, I vote AD to pre-sub is involved in head direction, but AD to RST is the contextual encoding because AD itself does a whole bunch of other things.
Hello, Ying and Daraj. Can you hear me?
Yes.
Yep.
Hello. Oh, very beautiful work. I actually have two simple questions. I might miss something. So first question is, here I think you identify the anterior thalamus, the dorsal part, the ventral part, and especially the dorsal part and their function in cognitive deficit. So first question is, what's your rationale why you choose schizophrenia and also and ASD as a model to test their functions? It's the first question. And the second related question is, here you mainly use the neuropsychiatric disease as a model. How about neural degeneration diseases? Have you tried their function in like AD or other dementia diseases?
So very good question. For the first one, yeah.
Yeah. So what's your rationale? You choose schizophrenia and autism to study their function.
Right. Very good question. Step one, please remember we are in a autism heavy lab. So for some reason, Goping loves autism. Do I love autism? Most likely not, but he loves autism. So part of the reason for studying autism genes was just out of luck. So Ying introduced PATCH-D1, right? So actually, as it became clear for the seminar, we started this project just as a traditional basic science study because not much was known. And then in order to learn more and to maybe elevate the impact of this study, we turned to diseases. When we started to screen disease models in the lab that the lab had studied, which have cognitive deficits, the only one that had very robust expression was PATCH-D1. And then Ying mentioned briefly, but the lab's PATCH-D1 paper in these knockout mice, they have robust contextual memory deficits. And then when they rescued all sorts of different proteins and channels in a different thalamic nucleus, it could not rescue the learning and memory, right? So that was, we saw it as a beautiful entry point. We have a reason now, wait, there's a question in the field, it's our previous work, it makes sense, and nobody answered the question. So when we started to dig deeper, we actually found out that removing PATCH-D1 just from AD leads to memory deficits. And so that was our first almost evidence that maybe it's worth studying diseases. And then after that point, because we're already doing an autism gene, we picked two other genes, for example, right? She already went over them, and we tried to look for ones that have similar converging effects. In parallel, I think it's very important. We wanted to, at the end of the day, I don't think anyone, or at least for us, okay, our goal was not to say AD is important for cognition in autism. right? Because many other diseases also have cognitive deficits. So we were maybe aiming higher. We wanted to see, can we try to elevate this work even more by looking at maybe a different disorder? And so one good example was schizophrenia, and let me tell you why. We work very closely with the Broad Institute. The Broad Institute in Cambridge, in Massachusetts, actually just released the schizophrenia database in the past 10 years, right? And they updated this database last September, or 2 Septembers ago now. So when this project was ongoing, they released this new set of really top 10 schizophrenia genes, which are highly correlated to patient symptoms. And among these top 10, there are only 3 genes that have, some link to synaptic deficits, for example, or NMDA AMPA receptors. And the cool part is all 3 genes are expressed heavily within AD. They're also expressed in other parts of the brain, but 3 out of the 10 that are linked to some way or form synaptic deficits were expressed in AD. And so that was our decision making at that time. We thought, great, we think 2 disorders is already beyond what we would do in the past, and it should make the point clear if we're able to, and that's why we chased 2. And to be honest, if we added any more in this study personally, I think we would have no space. And now coming to your second question, we did not do this in neurodegeneration models yet, just by time and effort. We had to choose some, but it's a very good question because ATN has been linked to a lot of, for example, neuronal death in Alzheimer's patient tissues. So what you're asking is highly relevant. We have not done it. If we have time, we'll do it in the future, and hopefully no one beats us to it.
Okay, makes sense. Thank you.
Other questions?
Hi, I have one question. So if I remember correctly, the AV can project to RSC inhibitor neurons, and it can modulate the memory specificity, right?
Correct.
Yeah. So did you also check the anxiety level? Like if you inhibit them, whether it's just like memory related freezing or it's just overall anxiety?
Very good question. For almost all the manipulations here, unfortunately, a reviewer troubled us to do all kinds of open field and anxiety tests. So you name it, for almost every optogenetic experiment, we have to test these, and we don't see any major manipulation. So long story short, if you manipulate AV circuits in any way, whether it's channel reduction, whether it's inhibition, no major change in pure locomotion or road rod behavior or OMAs that we can tell. It doesn't mean it does nothing, but at least following the traditional paradigms, we don't see a big effect.
Yeah, that's great. So for the memory specificity, how do you have any like hypothesis or guessing about the circuits behind it? Like how the inhibitor neurons contribute to the manipulation?
Yeah, we spent a lot of time on this actually. If you're curious, there's a nice discussion paragraph that I'm very proud of. We spent a lot of time, but let me just discuss it quickly. The idea we ended with in the paper was that, let's just think about a normal mouse, okay? Many brain regions are activated when you learn something new. Okay? And now if you just think about AD for 1 second, our model is that AD neurons have to be more active, right? When you're encoding context information. This activity obviously has to go to retrosplenial cortex because we already showed that's the downstream target. And then retrosplenial cortex neurons have to be active, right? And we know this because if you just inhibit retrosplenial cortex, you can block simple things like context sphere learning and many other contextual learning paradigms. But when you inhibit AV, what you see is that the retrosplenial cortex neurons are actually hyperactive, or you get a much larger ensemble, right? So I'm not saying that this is a complete explanation. But our idea was that the AD inputs to RSD help elevate, the activity of RSD neurons that are recruited for one learning experience, right? So a learning-induced increase. But any learning-induced increase has some range, right? Let's say 10% becoming 30% of neurons are active. When you take away AV, this 30% now becomes 60%. So our hypothesis is that it's not it's not preventing RST from firing. That's why you still get normal encoding. But it uses so many more neurons that now maybe there's, a loss of, for example, there's more noise in this system, right? Because you have so many more neurons than you need. And that's the at least the model that we try to propose in the discussion, that for any learning event, you need a specific learning-induced increase in neural activity. And in RSC, I'm sure there's some range. We think AD contributes to bringing this, RSC neurons to the range. But when AV is taken away, it's well beyond. And that's why the animals generalize. They just think every context is fearful. And we still think these neurons are manipulating, for example, inputs to entorhinal cortex, right? Because at the end of the day, we think all of this is routing to the hippocampus. So this is not our work. The previous work has already shown if you hyperactivate different subdivisions of the hippocampus, you can, in some cases, get almost a very similar generalization phenotype. And so that's our idea, that if there are too many neurons from entorhinal cortex, for example, we didn't show this, but this is our prediction, projects to the hippocampus and they're hyperactive during learning. It doesn't prevent learning per se, but then the specificity of this context is not as robust in the animal's brain. It's not a complete story, but that's the model we left with.
Yeah. Thank you. Very clear. I have one quick question. One last quick question is about C1QL2, the mouse line. If I remember correctly, C1QL2 is a specific marker for granule cells, like DG granule cells and cerebellar granule cells. Is the AD cells are also kind of granule cells?
Good question. They are not. So if you go look at the Allen Atlas, right, the in situ database, exactly like you said, the top region to where C1QL2 express is DG and basically granule cells, and then as you said, cerebellar. Sorry, I did not pay attention. But the second, right below that, is just AD. The rest of the brain, if you've looked, it's almost sparse labeling. We don't know why that is, but maybe you're right. Maybe for some reason AD neurons are in a way maybe similar, by transcription signatures. It's possible, but I can tell you they don't look or fire anything like a granule cell. But yes, this marker is the only common thing that I know of. But very good point. Yeah, it is expressed heavily in the dentate gyrus.
Yeah. Thank you, and very nice work and presentation. Thanks.
Okay, let me ask a question. So it seems the hyperactivity is a phenomenon of the cognitive problem. Have you tried to mimic this phenotype by enhancement of the neural activity by optogenetics or chemogenetics?
Good question. We did not. We did not for sure. We could have, just not something that crossed our mind. But theoretically speaking, our prediction would be that it should disrupt. At least if you make the neurons hyperactive before the learning, right? Because that's something that we addressed in the paper. There should not be a learning-induced increase. So there should be hyperactive almost saturation, right? So as long as you make the AD neurons with opto or whatever technique, it doesn't matter, to the, let's say, ceiling level before the learning event, our prediction would be that you should block the subsequent learning-induced plasticity increase, and that's why the animals don't learn as well. But we did not test it directly.
So this hyperactivity,
our indirect way.
Yeah, I see. So this hyperactivity is the property of the neuron itself. It's not because of the input or connectivities, right?
I mean, I think it's very hard to say, the inputs have no role, right? I don't think anybody can say that until we actually check. But what we can say is that at least part of it is because of the neurons themselves. Their intrinsic property changes. That's correct, right? So I think saying the inputs don't do anything, we don't know. But what we can say for sure is that at least part of the problem must be from the intrinsic change, because it's really black and white.
I see.
But yeah, good point. We did not look how much the inputs contribute to even maybe the establishment or formation of this hyperactivity. That's correct. It could be that, you know, maybe when you knock these proteins down in the first few hours or days, the inputs somehow also are changed. It's very possible, and I don't think we can rule that out. But yeah, very good question. Yeah.
I see. Is there any connectivities between the AD neurons and the AV neurons?
No, zero. In any direction, zero. And that's it's not. I mean, when I first came to the thalamus, I was really surprised because in other brain regions it's very common to have interconnections, but in a lot of thalamic nuclei, this seems to be the case. Minimal interconnections within, almost always out, and they always are very. Another property is this convergent. Many thalamic nuclei will lead out in the same cortical site, but they don't project it directly to one another. And I think for me, that's what's exciting. AD and AV are neighbors, right? There's a 5 micron distance between them, and they both converge in the same site, and they do almost 2 different things, but part of the same behavior.
Yeah, it's very cool to see the sharp borders, 2 areas. Yeah.
Correct. We recorded, and there's no. I mean, this is not new. You know, we need to give credit to all the people before us. It's already well known. If you stimulate an AD neuron, you try recording, there's no reaction, no response in either direction. Yeah, so we do think all the explanations have to be about the downstream, how the neural activity downstream forms or communicates.
I see. Okay. Yeah. Other questions?
Excuse me. I have a question. Maybe I missed something. Have you ever checked those autism or schizophrenia gene expression in ATN? Hello.
Can't really hear you. Can you repeat the question?
Maybe I missed something. Can you hear? Have you ever checked the expression of those autism or schizophrenia genes in the ATN?
I mean, so the ones that you saw in the presentation, right? I hope you may have missed a slide, right?
I mean the expression of those genes.
Correct. Before we picked which ones to knock down, we actually had to first confirm they're expressed in the ATN. And just from this slide, if you can see, among these 8, for example, among many more, the AD subdivision, if ASD and schizophrenia risk genes are expressed in ATN, AD is always common, right? I hope you can appreciate that. You will see many genes which are missing from AV, but they're always in AD, right? And we have a really nice plot in the supplement. So if you're interested, we think that's why this convergent idea may be, at least partly cool for AD.
So my question is, in those disease models, does the expression of these genes, the expression level of those genes change?
Yes, yeah. I mean, we tried to knock them down.
I mean, not in your knockdown, in other assays or other models, whether the expression level of those genes change in ATN.
Oh, I see. From what I know from the literature, I haven't seen anybody report it.
Okay.
Yeah. Sorry, I haven't seen anyone say that. Maybe it is. We have not looked in some other models. Yeah. But at least it's not on PubMed as of today.
Okay, if there's no more questions, let's thank today's two speakers. And if you have more questions, maybe you can contact with them offline. Good night, good morning, and yeah, everywhere, friends. See you next time.